Welcome to Neurosalience, the OHBM podcast.
Welcome to the Organization for Human Brain Mapping Neurosalience podcast. I'm Peter Bandettini, and here I discuss brain imaging and modeling with scientists from around the world. Today, my guest is Dr. Vince Calhoun, who's also a longtime colleague and friend. He's the founding director of the Tri-Institutional Center for Translational Research in Neuroimaging and Data Science, otherwise known as TRENDS, which is a consortium formed by Georgia State University, Georgia Tech, and Emory University. Vince received his bachelor's in electrical engineering from the University of Kansas in 1991, 2 master's degrees in biomedical engineering and information systems from Johns Hopkins in 1993 and 1996, and his Ph.D. in electrical engineering from the University of Maryland in Baltimore County in 2002. After 4 years at Yale University, he became president of the Mind Research Network and distinguished professor at the University of New Mexico before he moved to Atlanta for his present position several years ago. Vince's focus over the years could be summarized as using fMRI and other neuroimaging methods while developing processing methods to extract every possible useful bit of information. He's been prodigiously engaged and productive for over 20 years, advancing multimodal brain imaging, data fusion, and machine learning, among many other things. His work has inspired new ways of looking at the data. In this discussion, we talk about his work, his professional journey from the East Coast to New Mexico, and now back to Atlanta, as well as his successful battle with cancer in about the year 2010. It's always great to talk with Vince. He's extremely personable and just inspiring. I really enjoyed the conversation. I hope you do too. And here I have the great honor to have a chance to talk with my friend and colleague, Vince Calhoun, who's been in the field for quite some time, and you probably all know who he is. And he's been pioneering how to use and analyze and delve into the data of fMRI and other modalities and fusing it all together to make sense and draw insight. So thanks for joining us, Vince.
Yeah, thanks for having me. This is it's a pleasure to be here. So
there's a lot that I wanted to talk about today, but I usually kind of just start a little bit with some background in history. So I think I first met you really somewhat early on, but I think you were still in maybe still in Baltimore or somewhere in between before you went to New Mexico, and you were already very much focused on sort of the theme of what you're doing. You're sort of developing. I think you had just developed Group ICA and sort of put that out there. But so let's even back up further. So how did you go from focusing on engineering, which is your background and which you're still very much in that domain, that field, to brain mapping? How and were there any decisions along the way? Any mentors? Any defining experiences?
Yeah, so I guess I was trying to think about that a little bit, and I was I guess I don't know at what stage you want me to start, but I think I've always been kind of curious about how things worked, and I was kind of your classic nerd in school, and I also liked kind of early on like to take things apart, put them back together in different ways, and sort of combining technology and math and discovery all together was kind of an exciting space. So that was kind of the stage, I guess. And then, as you mentioned, I did my bachelor's degree in electrical engineering, and was really, not biomedical at all, at that point. And it was probably in the last year of my bachelor's when I started thinking about graduate school, where kind of two things kind of came together, where I just kind of, I don't know, it was like it was a magazine article or something where it was talking a little more about MRI. And so I got really, I thought, this is really cool. You're manipulating magnetic fields and you're getting an image from that. And so I started reading more about that. And then in parallel, I also started to become more aware of how much you can model the human body using engineering principles. So, things like electricity, right, in your body, neuronal firing or flow, fluid flow, blood vessels, all that stuff. And so that kind of brought those things together. And so then for graduate school, I started looking at, more sort of biomedical, electrical type, programs. And so that then led me to, I ended up going to Hopkins for biomedical engineering initially, and so was working with Cam Leong and doing some like early diffusion. It was like a contrast-based diffusion thing where we're putting polymers with gadolinium DTPA in rabbits. So I was doing animal surgery, and wow. I built a couple MRI coils, and worked on a GE Omega 4.7 Tesla scanner. So lots of memories of we did it always after midnight because that's when it was cheap. Lots of discussions about, is the rabbit still alive? Tuning the coil profile with the long stick. All of that stuff. And so, but that was, as kind of right around the time that fMRI was being discovered. Sort of early 90s.
Yeah.
And I ended up, I think I did, after 2 years, I finished with a master's at Hopkins, and then I started working at Hopkins in Godfrey Perlson's group in psychiatry.
Oh, wow.
And sort of, so it was still brain, but it was getting more into neuropsychiatric, space. We started out with structural MRI, but I got very interested in fMRI, and I started pushing to get the group more into fMRI. And that was, I think I went to the early Boston meeting, and they also had like, I think, a week-long course that I took, and got kind of into, really into fMRI. I was trying to get Peter Van Zyl interested in activation studies. He wasn't initially, because he was more into the metabolic and the spectroscopy, still very engaged in all that early work, of course. So that was, so that was a lot of fun. And
that was also at Kennedy Krieger for the most part, right?
Yes, exactly. Yes, exactly. So I think I gave a talk at the opening session there in, I think it was 99, on some latency fMRI stuff we were doing at the time. But yeah, so there was a lot of ongoing activity and a lot of people that were interested in getting into that, learning more about fMRI. So, but I was with Godfrey's group, I was doing a lot of programming. I was like a programmer with them. So I did, we were building, sort of morphometric programs for measuring volumes, stereotaxy, things like that. But then I started in this side thing, getting into fMRI, and that was really what took off, I think, for me, is really, kind of this really sort of exciting area. Yeah. And then I saw Martin McCune's paper in 98 on ICA, and I think they had like 4 subjects or something in their paper, but they had a whole bunch of, it was a pretty detailed paper about, working with ICA. And so then I started playing with that, learning more about that. And that's when I started focusing a lot more on ICA.
And it was cool about during that time. Yeah, no, I also, Jim Picard probably was at the group as well. And I also remember at that time too, right? Early on, that's what was so fun about people who actually had well-grounded training in, signal processing or things like this. Basically, fMRI was sort of like brand new territory.
And
by
the way, so I did my master's in biomedical, then I worked for Godfrey, and then just a really brief thing, and then I did another master's in information systems because I was doing all the programming stuff. And I didn't start my Ph.D. until later. which I did kind of part-time at UMBC while I was working at Hopkins. But I don't know if you remember, I was looking up this as we were doing this, preparing for this interview, but I think in 1996, I emailed you about the MCW BME program. And so you sent me back some information about it at the time. So we actually connected, sort of way back then, just at that level. But yeah, it was kind of interesting. I'd forgotten that. So.
Yeah, I had forgotten that too. Yeah, interesting. Okay. Hopefully I was helpful, but
yeah, you were.
But it's a good thing. Yeah,
you didn't blow me off. So
yeah, back then I was actually, I just probably had started at MCW as an assistant professor after doing my postdoc, but still, things early times. Yeah.
Yeah. No, I spent a lot of time with your 93 paper too. Just that was fun.
Yeah. Well,
so, yeah.
I mean, once again, that's a classic example of basically, second year college sort of, sort of processing sort of applied in a new way. And it was like, hey, you can do correlation. But yeah, I mean, you picked up on that theme incredibly well. And so, I mean, your first, okay, so let's, maybe also before we get into some of your work, I mean, then, just to trace for the sake of completeness. So you finished up in Baltimore and got your PhD in 2002. Then you went to Yale briefly and then went to New Mexico, where you built a great place, and yeah, you can go on from.
So I moved. I went from I was with Godfrey about 10 years, and then he got a position at the Institute of Living to create a new imaging center there, and he brought me along to help build the imaging center and continue the work that we're doing. And so I jumped at that opportunity and went up with him to Yale and Hartford Hospital. Okay. And I was there about 4 years.
Okay.
And then the Mind Research Network and the University of New Mexico opportunity came. And that was, for me, I loved working with Godfrey, and it was, I was sad to leave him, but it was sort of an opportunity to continue to grow and expand. And as you know, I've continued to work with Godfrey for many, many years. So this was a center that it was actually already built, right? So then I was able to just jump into that and basically take on that role it was very well suited for me and a good fit. And I was there for about 12 years. And then this opportunity with Atlanta came about, where they were very excited about, allowing me to create this tri-institutional center between Georgia State, Georgia Tech, and Emory. So it's very collaborative. There's a lot, of, it's just the barriers between institutions can be challenging to navigate. And they've been able to, better than any place I've been at, figure out how to navigate those. So it's been great coming out here. So anyway, that's sort of the brief nutshell. So for me, it was typically a pull, not a push. Like I was like happy where I was. And then this new thing was like, oh, that's really, an opportunity. And it was hard to say no to that. So
yeah. And it seems like every place you go, it seems like you ratchet it up a little bit more. And yeah, what I've been down to Atlanta, and it's super impressive. And the amount of people that you have and how you're coordinating it is, like you said, I mean, it must be a huge challenge, and it's nice because, you have, this should be done more often, where you have sort of more consortia between universities sharing resources and knowledge and information. And yeah, it's great. So just to maybe start off, briefly with, taking a few steps back. And I think at least where I first remembered your work, and you mentioned it briefly, was, starting to work with ICA, which is the, a next step beyond, things like correlation analysis and whatever. And it seems like your unique contribution, if I recall correctly, was figuring out how to do this in a group level, I guess, sort of like group ICA, and which also seems, all these things that work out well were not obvious at the time. I mean, everyone's sort of struggling with things, and then after the fact, it's like, oh yeah, that makes sense, and as far as that's concerned. And I don't know, as far as that, I mean, people still do that today. I mean, they do many other things, but so it's sort of caught on. And so maybe just one question about that: is there, people were playing with PCA at the time, and PCA, the problem with PCA potentially was that there was a little bit too much. It was hard to really confidently unmix the signals, the artifactual signals from the real signals in time. And it seems like that ICA did this better just by creating a necessity that the maps were spatially independent, which actually seemed to work better in separating artifact from actual signal. And of course, there's much interpretation that goes into how many bins you choose and where you cut it off. So what was your rationale? I mean, maybe that was the rationale, but you were trying these various data reduction techniques and ICA seemed to work better. Was it more like a bottom up, or was there a principle, or what was your, if you can recall?
Yeah. No, I think for me, I was playing with a lot of different sort of decomposition based approaches, pattern based approaches. The thing that I really liked about ICA, I guess, was this larger context of blind source separation.
Yep.
Which is very general. You can do a lot of things with it. ICA is a particular type of blind source separation, and the fact that it uses higher order statistics. So PCA is great, but it's focused only on variance.
Yep.
And so separating, pushing all the variance into one component it doesn't, as I think, in terms of just visualizing what's going on in the brain, in particular brain networks and things like that we're looking at so commonly now, it's not really focused on distinction between these things. It's really focused on maximizing variance, right?
Yeah.
And so that use of a higher order, statistic that is basically general independence, not just independence of a Gaussian, has turned out to be really useful. And I think initially it was just playing with it, and it's like, oh, these look kind of cool, they look kind of interesting. So it was just this sort of, this looks interesting, and then kind of going further and further down the path. And I think that the thing I noticed too about some of I think there was just McCune's early work really was the main thing that I saw for fMRI. And that was, I think they had like 4 subjects and they just showed them side by side. Yeah. And I'm like thinking, being in Godfrey's group, we're interested in, psychiatric conditions, and we want to look at, comparing across subjects. And so the idea was not really to get a group map. The idea was to create a group inference framework that we could use to have correspondence between individual subjects. Yeah. Right. So that first paper that I put out on the framework was really, a lot of it was about back reconstruction and getting individual subject estimates out from this larger framework. So,
okay, but it is interesting how, I mean, you kind of suggest, I mean, this is sort of leads into almost everything else that you've done, and there is sort of this art between, coming up with analysis approaches and sort of getting a sense of what the data and the noise are like, and also what the variance of the noise in many different ways, the time series noise, the subject variance from subject to subject, and all kinds of things, and actually getting a sense of what aspects of the signal that you wanted to separate, and it seems that that's kind of the name of the game for what so many people are trying to do. I mean, even today, we're still trying to model the noise better. We're still trying to pull out every last bit of information from the time series signal, whether it's spatial correlation or latencies or undershoots or things like that. So it's like we're always trying to sort of guide it with rigorous techniques, but at the same time, sort of building our knowledge of what the separate things are. So, and you've been doing this incredibly well. So, so I'll just keep on going as far as that's concerned. And I will maybe get into a little bit in terms of group normalization, the problem of, it seems like it's arisen in the context of, at least for me, the BWAS paper that kind of came up, like that said, well, you need 2,000 subjects to draw any sort of inference from resting states. And then, why is this? And can you do better than that? But before I get into that,
I don't think that's what the paper says, but
no. Okay. You're absolutely right. I mean,
but I know I've heard the session you gave on that. So I know what you mean. Yeah.
No, it's actually, Right. I mean, it doesn't clearly imply that. It says that done a certain way, things start to
Yeah.
stabilize at 2000.
Effect size, cross-validation, things like that are important. And below a certain value, your sample size has to go up. Yeah.
Right. But I do think at least for me, what that has a lot to do with potentially, obviously, there are many, and they even said this in the paper too. I mean, there definitely, it was a great paper, and they said that there's heterogeneity across the populations. There's that might reduce the effect size. There's heterogeneity of brains that putting things into a normalized brain and then parcellating it against a template, I think throws away a lot of the interesting variants that might exist as well. And that's always a challenge too. And also resting state itself might not be the most powerful technique for pulling out differences. And so, we're trying other things like movies or tasks, but that's another issue.
That's why I'm not a fan of parcellation as a first step. Like I think the other thing I really like about IC8 is that we usually work in component space, right? So we don't work in ROIs or thresholded things, right? So they're whole brain patterns that overlap with one another. Yeah. And so you're retaining all of that information, which can be really useful you can think about how certain brain regions, probably all of them, do more than one thing, right? So allowing your model to allow for that flexibility, I think, is really important.
Yes.
So I think I would completely agree with that.
And that's a really good point that I think a lot of people may not realize. Right. I mean, starting out in sort of that's parcel space, but network space derived by ICA or other methods that are more bottom up, sort of from each subject, I think that gets around that problem really nicely.
And I think another thing too, just to add to that, is this is kind of how I think about stuff a little bit. Is your starting point, you have to be really careful about your starting point. And so that's where at least I've spent most of my time, right? Everybody has their different focus, right? So, which is this space that we're pulling out of the brain that we're then analyzing. Is it reflective of the complexity of the data?
Yep.
And for example, we do a bunch of things where we don't just do ICA, we maybe have a constraint applied to it. And that changes your ICA components. But for example, when we talk about the dynamics piece, things that are good representatives of static activity may not be good representatives of, they may change over time, right? Yeah. And so having adaptive functional unit type methods, I think it's still underexplored.
Yeah, I agree with you. I completely agree with you. Right, because I think people are. We really are. I think the field is struggling about. Like you said, even the starting point. How do you start to even think about? Some people are very much tied to sticking to tasks and knowing what the timing is. You have to know certain things like the timing if you're doing things, and what their behavior. They try to measure the behavior as well as possible and put that into a space. And right. Generally starting in spatial normalization space and then trying to average first is, like you said, it's
and task data is really important, I think, too. And you see the components that you look at if you put them together.
Yeah. Yes.
You see they vary. They're a lot of the intrinsic network information is still present. It's not like you have a completely different brain when you're doing a task, but it changes. Maybe it grows or shrinks or moves or things like that. So
and so much of it, right? So much of it is information, whereas I think people quite easily look right beyond it towards what they think they're looking for and miss that sort of subtlety. But
hopefully we'll all complement each other's biases, right? So
that's the goal. That's the goal, and hopefully it's working that way. So I guess moving ahead to about 2000, the early 2010s or to 14, somewhere around there, you sort of coined a term, the chronectome. I guess it's probably not absolutely new. I think it's been used in other contexts, but Could you describe what that is? And sort of like that was right around when dynamic resting state sort of was coming out, and seems like you've picked up on that and kind of ran with it and took it further. And so if you know, could you talk a little bit about that work?
Yeah.
Yeah.
So this came about from, again, recognizing that we're always looking at functional connectivity, or we use the term functional network connectivity a lot, which is really similar. It's coupling between time courses, but
Yeah.
we're working with whole brain overlapping, components. That's basically component time
Yep.
courses. So
Yeah.
a component is a functional connectivity network.
Okay.
And we're looking at the interaction between those things. But
Yeah.
yeah, I mean, I think recognizing that, we're looking at average connectivity over, say, a 5, 10, 15-minute run, but the brain is not sitting there doing nothing. And so, while we can analyze it with tasks for rest data, we typically have just been averaging, over time. So kind of starting to get into, do the patterns of, connectivity, vary over time in an individual, and can we leverage that information? Can we allow that information to be captured in the approach? And that's really what that term chronectome was used to describe, was a real general sort of whole brain framework for recognizing that there's time varying connectivity that we should be capturing.
Yeah. Yeah. And it seems that, I mean, you did a few things. And I'm kind of curious how they've gone, since I haven't been able to even keep up with everything that you've written in detail. But so, like, for instance, the one thing that really excited me about the connectome initially was, it's almost an iconic figure now, in which you were differentiating between those with schizophrenia versus normals, and you were showing the amount of dwell time in certain states.
Right.
And I was, super intrigued by that, and thinking, okay, you can do all kinds of things. If you characterize the states, you can actually maybe look at the sequence of states. You could look at the dwell time. You can, all kinds. So you've probably continued to try to push that. How has that sort of panned out in terms of,
Right. Yeah. I mean, I think it's, we're continuing to explore it. It's continuing to be a very rich space. I think that first paper looking at schizophrenia it's one of my favorite examples, like you said, of why we might want to do this, because it really, I think, illustrates that there's more information than just in the average. Right. And so we found these sort of more modular states that were, individuals that, the controls spent more time in the more modular states, and then the patients with schizophrenia spent more time in these less modular states. And then, we've continued to kind of push that, where you can look at transition times. You can look at, there was a follow-up paper where we modeled the states as, so you don't have, it's again still simplifying to say the brain's in a state, right? It's really more realistic to consider that continuous representations of these things over time that overlap with one another. So that was a follow-up where we looked at with some work with Robin Miller, this sort of state space approach, where we have, a given coordinate in that state space is a multidimensional vector, which we call the metastate. And then we can summarize those and look at, like, how much distance do people travel? And the thing I like about the coolest thing I liked about that paper was that this definition of what she called hub states, which were kind of like sticky states, right? So once you get into them, you kind of get stuck for a little while, and you stay a little bit longer. And the patients showed much more, much more prevalence of these sticky states, and that the time they spent in those sticky states also was related to negative symptoms as well. So it's kind of, it's sort of, again, we're looking at this stage just correlation, and there's more work to be done, but it was really an interesting sort of, for me, example of how we could use this information to get a little bit deeper.
And yeah, it's interesting also that time scale, like, the states last maybe anywhere from 30 seconds to, a couple minutes or whatever, is so informative. It's kind of like sort of the time scale of how people's, thoughts drift in some sense. And
Yeah.
so,
Yeah.
but it'd be nice to even go faster. I mean, we've been trying to pull out, more instead of using like windowed correlation, which seems problematic. We and other people have been trying to do things like, more instantaneous correlation, like, it's equivalent to like the co-activation pattern studies. We've also been trying to do edge analysis, and you can pull out these what looks like bursts of correlation that occur at a faster time scale. That's also really informative. So there's more to be done in this space, I think.
Yeah, and I think I would also frame it. I mean, so we've been doing this sort of instantaneous stuff for since the early days as well, and it is useful. I think the thing I would frame it in terms of, because I do see a lot of like papers, like windowing is kind of an obvious like straw man, right? So you can say, you've got a window, that's bad, do this instead, right? And I think that's probably not the most useful framing, because for example, you can think of a window as simply a choice of filter on your data, as you know. And you can go like some work with Ashkan Fagheri here. He created basically this framework using filter banks, where you can essentially design an arbitrary set of filters, and you can cover the full space or some combination of that frequency space, including instantaneous, and you can unify it with static. And I think it's just really interesting. That's kind of where also I think it's nice, because then you can combine it with the prior work as well. And essentially that's really what we're doing with these sort of chronectome type analyses, is we're unpacking the static analysis, into something that's a little more time resolved.
Yeah, and definitely, I think, and you've been one of the pioneers for this, and I'm always curious to see how you're taking this, and it seems like like you mentioned, there's so much more to do.
No, really, and I think getting at temporal resolution too, I think would be other modalities, right? So EEG, MEG, things like that, which there's a lot of ongoing work in that. It's a really I think useful way to model the data. I think when I think of fMRI and EEG, for example, and if I think of like cellular information and network information, we're really talking about multiple spatial and temporal scales, and so we're thinking a lot more in that way. And at this sort of macro scale, either in space or time, you see patterns that are, I think, informative. They're not telling you about that very fine grained scale, at least not in excruciating detail. They're telling you something about how it manifests at this other scale. Right. So I think there's a lot of interesting work still to be done in sort of bridging spatial temporal scales as well.
I completely agree. And I think, there's only like a handful of people actually really doing it in sort of detail. And your group is certainly one of the pioneers as far as that's concerned. Maybe we might even get into this part instead of to go on into the whole idea of data fusion, of multimodal data fusion, which this is just one aspect of, and we can go back to maybe trying to talk about individual assessment and clinical applications, but multimodal data fusion, you had a review article in 2015, and I realized that, probably most of your work now, most of it, I think, has been along those lines of trying to combine, structural, anatomic, genetic, EEG, fMRI data together in some way to draw out synergistic information or complementary information. And you've really written nicely about how it's not just simply, one directional, like MEG or like fMRI might constrain the inverse problem with EEG, which is kind of works, but doesn't really work great. But it's more like a two-way sort of thing. And I'm kind of curious, what has been your intellectual journey along that line? What's worked well? Yeah.
Well, I think, like you said, I've been doing data fusion a long time, and I really like it. I think there's a lot of untapped information. And one of the things that really convinced me of that was this, I think it was a 2006 paper that I wrote, which had neural chronometry in the title, I think. And so we were using fMRI and EEG data, but it was separate sessions. right? So we don't have the time information linked like you would in a concurrent acquisition, but we had multiple subjects, and so we can link at the subject level. And so we had these event-related potentials that we were looking at, coupling with the fMRI event-related activity, and so we fused it at that level, right?
Okay.
And so we have this joint components that we get out. But what was really cool was then we could take these joint components, and we can take, for example, the EEG signal, which was split into several different components that comprised. We're looking at the P300, and so it had these early N1, N2, and P3 peaks. And so we could take those components that were joint with an fMRI component.
Yep.
Each one had a pair, and then we could pick a period in time at the EEG temporal scale and reconstruct an fMRI map.
Nice.
With a combination of those, right?
Yeah.
And so it's kind of like cheating, right? But we're basically moving along at millisecond temporal resolution and looking at fMRI combinations at that millisecond temporal resolution.
Yeah.
And it was beautiful. It gave us this really nice mapping. This was an oddball task. So you have a sound, and then you have motor response if it's a salient response, and a button click. And we saw in the fMRI map, we saw auditory cortex, then we saw SMA, then we saw primary motor. It kind of came on in the order you would have expected. And then this big P300 response in frontal lobe, kind of more globally.
Yeah.
And that, to me, I was like, wow, that convinces me that we need to be doing more of this, because how can we do that? How does that work, right? And so we're leveraging, again, this linkage between two different modalities, and we're looking at this space where you can leverage the strength of both modalities and bring it together.
Yeah, and just to emphasize, it's so much better doing that than trying to solve the inverse problem in terms of EEG, to sort of like localize where the dipole sources are, since it might give you a map that might look a little bit like that, but the fMRI is so much more.
You can do both, right? So you could also do EEG source space, fusion with fMRI. You can do the reverse, where we can pick a voxel in fMRI and reconstruct a time sequence from the EEG, right? So you're moving around in space. And so, that kind of stuff, it's for me, it's like, I've always got that in mind when I think about data fusion. It's very, it's a very powerful example, I think, of how that can work. And like you mentioned earlier, kind of one-way symmetric, that's a sort of rubric that I like to think about, in that they're all useful. So, there's a spectrum of fusion, right? And the simplest one is I just do a structural analysis and a functional analysis, and I look at them and I put them together in my head. The second level would be, maybe I want to co-register them like a PET CT fusion. So they're overlapping. The one-way analysis would be, I've, maybe used, like you said, fMRI to constrain EEG. And then the symmetric analysis, where they jointly inform one another. So,
that's actually, that's a beautiful way of putting it out. And it reminds me a little bit. I've been bugging and maybe you've already been trying to do this as well. And it's not the most original idea for me, at least. I mean, it was sort of like a thought that I have Pete Malfeas in my group does EEG and fMRI simultaneous. And I keep on thinking, let's make a database of relationships between the EEG patterns and the fMRI patterns, just anything, like with, various tasks or whatever. So we don't even need the fMRI. You can actually, completely inform someone, walking around with the EEG to, what the fMRI patterns are. And so it's kind of ambitious and it's kind of open-ended, but
actually that's really cool. I think two examples I have from that are, one is, remember the early Barat Biswal's resting state composite data set, the big one.
Yeah.
And he asked me to contribute to that, and I didn't, and I still am like shooting myself. But I think there was a follow-up study that looked at some EEG fMRI concurrent data and estimated sleep measures from EEG, because those are well known.
Yeah.
And used those to develop an fMRI predictor of sleep and drowsiness.
Yeah.
Then applied it to this data that had no EEG. And the inference was that, more than half of the subjects were drowsy or sleeping even.
Yeah.
Which, of course, they're asked to rest. So, of course, they're going to do what they're told. And so that's an example where you use one modality to inform the other, and then you can use that model on just one modality. And we've been doing a similar thing with behavioral assessments and imaging data. We're kind of looking at measures of trying to assess brain health and things like that. And so we have you can predict a future score on these sort of measures that are based on assessments.
Yeah.
And then you can also derive a sort of image assisted prediction model of that future score that doesn't require using the imaging data in new subjects, and it does better. So it's using the imaging data in subjects that have both to tell the model, well, ignore this, focus on this. That's the stuff that the imaging data tells you that the assessments doesn't tell you are important.
Yeah.
And so so that's another example of how you can use one, do a multimodal fusion with the goal of let's get to this cheaper or faster or more effective modality, informed still by the richer modality that's more expensive that you can't do in everybody.
Yes. Yeah. And that's right. That's a beautiful sort of practical application. Has there been any, so just along these lines a little bit more, I mean, has there been any, insights into like, as far as like, what information fMRI might have that we didn't see if we use another modality, we actually, reveal that it's there, or what has been the best or the most informative or insightful uses of it so far? Other, it might have been what you just mentioned, but in terms of clinical insight potentially, or because I think it's so useful and I think it's so much potential and I
mean, a recent example, we've got this model called parallel multi-link joint ICA. So it's again, naming these things is hard because you keep tacking on long words, but PIMJICA, whatever. But we have this. It's an extension of a joint ICA framework, but we're throwing in a bunch of brain networks and structure. And in the context of that, we're looking at Alzheimer's disease. In that study we did, and what we found was that the functional information and the structural information, we had these two sets of components which had a lot of overlap on the fMRI side in terms of a lot of default mode related regions, but they weren't identical. There was like thalamus and there were some different things but they weren't separated if you just did fMRI. And fMRI is, for Alzheimer's, generally the structural data tends to be a little bit more, it's like the reverse of schizophrenia. It tends to be a little bit more, because of atrophy, a little bit more sensitive to the disease progression.
Yeah.
But in this case, we split apart these two components, and the effect size was really high in one of them and near zero in the other one. And it hit the things that you might expect, like hippocampus and thalamus and other things like that. Yeah. And so that's an example where you'll have a more useful separation of essentially this information by leveraging the links between modalities.
Yeah.
In this case, we got lucky. It was more informative because we weren't trying to optimize for that. But that's just one recent example where it might be useful.
Yeah, that's okay. That's cool. Actually, so right. I mean, that's a trick. I mean, sort of like it helps sort of guide you in terms of where to look. And I mean, and there's this iteration that occurs, and it seems that to help with the iteration, you've also been, you've been like a lot of us have been trying to do, but not quite sure how to do it. Is embrace, machine learning and AI or whatever to more efficiently get discover these hidden associations that you wouldn't just see by looking at the data. So
yeah, absolutely. And I think what sort of a cool connection is, I mean, I see it, I would say, is machine learning. And the Infomax algorithm for ICA is a neural network model, right? So it's a single layer neural network model. And you can make it multi-layered. You can add, layers to it. There are different tricks you have to do to make it, work and be unique and all of that. But it has, it does have that potential. So yeah, I think it is there is a little bit of art. I would say also there's this whole circle of things, right? Where you kind of have, I spend a lot of my time in the data-driven space. right? But some people spend a lot of their time in the very refined, sort of model space.
Yep.
There's been a big push in recent years for computational psychiatry and looking at models. And I think that's great. And I think that helps fill out the gaps in the circle. But I also think it continues to be, it's the same, over the time you start to see things cycle, a little bit. And so, I see, again, clusters of people doing research, and they're still splitting up. Like there's focusing on, everybody's doing now resting network mediation models or something like that, or, and trying to answer interesting, computational psychiatry questions. But you're just looking at a small piece, and that's fine, but just recognize that's a small piece. And then the other side of the coin is just throw all the data in there and see what you can find, and make up a story, right? That's the sort of cynical view. But
right.
If you can do that in a way that maybe identifies some interesting relationships, and then evaluate that in a new study or new data, that gives you more confidence that that it's useful and real. So all these things have to inform one another, right? Otherwise, we're missing. Like you said, it's all about not knowing what you don't know, right?
Right. That's exactly right. I mean, I think people could easily criticize fMRI as a lot of the research is exploratory, but there's some aspect. I mean, the technology is changing so quickly. We're having new capabilities of combining it and looking at it in different ways. That there is always some amount of exploration involved, but as long as it iterates into sort of hypothesis-driven directions from there, you can imagine the process. But I think it's actually, and you've been doing this with your groups and with your number of groups and other number of people, all sort of coordinated so well in working on it. And then the trick is, so
you say, but yeah.
Well, I think it speaks for itself.
You don't know half the story. No, I'm just kidding.
I'm sure. Right. I'm sure that, right. You go into your group and say, okay, work harder. Okay.
No.
But then the other question is, the field itself is just humming along. And so we feel like we're making progress. And then there's the other side of it where, people are thinking, okay, well, when is there going to be, there's some hints that there might be signatures in clinical populations, and there's some insights into how the brain is organized. But are we really getting close to, using on single subjects, like putting a person in a scanner and saying, oh, you have this type of schizophrenia, you're most likely to need this type of drug or whatever on a daily basis? Do you think that we're actually getting closer to that goal, or even should it be a goal as far as using it daily?
I think it's a good goal. I think we are making progress in that space. And like you said, it's been slower than we would have liked, to get there. But, I think we're getting a lot better at, so the first step is just characterize what is going on with these individuals, right? And what's different about their brains. But the complicating factor is it's not a unitary type, disorder. So it's a syndrome, schizophrenia, for example, it's a syndrome, right? And so it's a mixture of unknown things. So your models have to try to account for, subgroups and mixtures and things like that. But one of the things I've really been excited about is we're doing a lot of work with keep doing ICA work. So it's work with the ICA where we're doing these spatially constrained models. So we have these replicable templates that we've derived from large data sets. So we do separate ICAs, and then we find ones that replicate. And we have several of these now, including this sort of 105 component multi-scale one that Armin Iraji's been developing. If we look at these in multiple separate groups of chronic schizophrenia and controls, you get very, very consistent patterns of difference, right? So, whether it's actually causing schizophrenia is a bigger question, and it's not what we can test given this data, but we see these consistent group differences and even consistent individual differences. We can do pretty reliable predictions of individuals, and we can say schizophrenia or not, right? That's not a clinically relevant classifier, really.
Right.
because it's easier just to interview someone, and make that decision. But it tells us these patterns might be informative, and can we leverage these patterns? And they're actually remarkably consistent with some of the early models of schizophrenia from like 20 years ago, right?
Yeah.
So it's subcortical,
Yep.
cortical, thalamic type things. So we're starting to look at these in a very data-driven way instead of coming at it from the other side,
Yeah.
and then asking questions about, like, can we predict response to medication?
Yeah.
I think it's really interesting. There's some new studies that NIMH is doing that you probably know of, where they're looking at sort of prodromal or sort of clinical high risk populations,
Yeah.
and they're going to have a decent sample size. In theory, they should have about 1,000 or so, and so that'll be a really rich data set
Yeah.
very early in the process that I think will help. Another
That's
bit
cool.
of work I'll mention that I really like. It was really interesting for me is psychiatric disorders overlap, right?
Yep.
And so one of the things we did was a recent study called a brain-wide risk score. And so what we did was we took 4 chronic patient sets of data where we have schizophrenia, autism spectrum, major depression, bipolar disorder, and we derived from each of those separately signatures of either controls or patients.
Okay.
And then we looked at the distance of those patterns to individuals in the ABCD study these are kids, they're not diagnosed, they're too young to manifest any psychiatric illness.
Yeah.
And then we characterized those. And it was really interesting because, again, we don't know yet what this really means, but the control, sort of 3 things that were kind of really interesting to me is that the control-like pattern was very obviously visible in at least half of the subjects of ABCD.
Yeah.
The patient-like pattern, if we say schizophrenia, for example, was only obviously visible if you went up to the upper 1% of risk in the histogram of these 10,000 subjects.
Huh.
And so it's a much weaker signature, but it's clearly there. Whether it means they're going to get schizophrenia or not, we have no idea.
Yeah.
But it also correlated with the prodromal risk score in the ABCD data.
Huh.
And so that was really interesting.
Yeah.
And then looking at, since we did all of these separately, we looked at the overlap between the different patterns in the ABCD folks relative to those distance measures. And we found the control-like pattern was very consistent across all 4.
Yeah.
The patient-like pattern was very diverse, right? So there were clear distinctions between them. And the sort of the upper, so we're looking at the upper, 5% and the lower 5% of this 10,000 subject histogram or so.
Yes.
And then the last thing we did was you can look at those subjects that fall into those tails and look at how they overlap. And so there were a lot of schizophrenia only, depression only, autism spectrum, but we also saw the most overlap between autism spectrum and schizophrenia and depression. So you saw a lot of folks that were in the tails for both, for high risk. A subset of folks that were in the tails for all 4, and so that's the kind of thing that I think we'll see over time. We might get more information. I don't know how much will emerge by the time ABCD is complete, because it's only going to go up to who knows? But for right now, it's going to go up till 18 to 20 years old. So, but it's the kind of thing that I think that's a promising direction, I feel, because we're not looking at categorical, yes or no. We're looking at sort of risk type measures and overlap and things like that. So,
yeah, that's actually really cool. I mean, right. If it could be a prediction or some sort of measure based on screening early enough, and not only will give you some sort of insight into mechanisms, depending on the features, right? I mean, you could actually look at the features that are most informative to really give you some insight into maybe potentially deep brain stimulation or other things might be interventional. And it's always tricky though, because you don't want to, always be have the risk of, this is always the problem with screening, is that you don't want to, have a
Make a
false
mistake.
positive and tell them, oh, watch out, you're going to get schizophrenia, which is
It's like
tricky.
self-driving cars, right? It's kind of like that. You hit one person and the whole program's gone, right? So I mean, I think operationally you can consider like a staged program, right? So it's like, this doesn't mean like, oh, doom and gloom. This is just elevated,
Yep.
a concern, and let's just look at it in a few other ways, right? So it's not yes or no. So I think that could be dealt with if it's useful, right?
Yeah,
So.
yeah, that's really cool. And actually, I completely agree. Along the lines of what you were mentioning is that so many people over the past using fMRI, it's like easiest to look at cortex, but I do think that fMRI and other techniques or especially fMRI would benefit from. I mean, I think all these disorders are somehow. I mean, I could be completely wrong, but it seems that they're somehow linked to subcortical links or processes that are somehow disrupted.
Shows up a lot. Yeah.
Yeah. And if they, the more we can actually go to high resolution or whatever to sort of tease that out, you might get higher quality features.
Yeah.
So,
Yeah.
Yeah. So, okay. So I mean, there's so much, it really is, you really do to get a sense that the field is progressing in a way that's meaningful. And hopefully it's actually, the horizon isn't always sort of receding. It's sort of like, hopefully we're actually getting to the point. And we haven't even talked about this. It might be another conversation of like, nuts and bolts sort of clinical implementation, just the practical stuff of actually being able to have a clinical fMRI suite in a hospital to be able to do that. So, I mean, I can't even imagine, a technologist sort of, having your array of multimodal integration tools and it has to be a clean pipeline that just, drop dead works.
Exactly. Yeah. Yeah.
Yeah. So in the last part of the podcast, I just wanted to, and we had talked about this beforehand that we were going to mention this, but I just remember, right around 2014, I was lucky enough to be asked by NSF to sort of travel the world and, I visit all the best labs in the world to write a report on the best labs of the world. And it was great. It was so informative. And I remember you were initially part of that,
right?
And then you had to duck out, and the thought was, we just heard that it was for health issues, but then then it came out, and you even posted on Facebook that that you had this pretty scary diagnosis. And I think it's important maybe to talk about this because I just had one of my really good friends, Sean Merritt, who just died of mesothelioma. A lot of people struggle with health issues, and I'm kind of curious. I really am. We never talked about this before, but I'm. What was it like hearing about it and then struggling with it and then moving on and getting over it? I mean, how did it affect you and your work?
Yeah, no, it's I'm as you mentioned, I'm open about it. I think so. It was I was diagnosed with lymphoma, and I had kind of in my gut area, I had some growths, and I had you could see it throughout my body basically.
Yeah.
And you saw, I think I posted the PET,
I'm like,
CT
oh my
fusion, PET CT fusion on Facebook, before and after. So I had a good after, right? So that was kind of a nice contrast to see.
Which is amazing in itself. I've never seen such how everything disappeared. It was just amazing.
I know. I was like, wow, that's awesome. So but it was that was 10 years or quite a bit later. And so I have, as I have 3 young kids. They're not so young now, but now I have a son who's 21 and my daughters are 15 and 13. Right. So, and this happened, I think about 14 years ago. It's like 2010. It was literally the same, within a few weeks of my youngest daughter being born.
Wow.
So my poor wife, is all I can say. And, so she was in the hospital for a few weeks because she had like a hole in her heart and things like that closed up, and it was kind of a sort of resolved itself. But then right after she came out, I went in, and I had some pain, and I went in and just had it checked out, and then they did a CT, and they're like, "Oh, we got to go do some more tests here," and then they diagnosed me again with lymphoma. I was very, distraught, I guess at first. And I was, you can't help but think, especially because my daughter was just born, right? I'm like thinking,
Yeah.
well, her name is Sarita, and I'm like, how am I going to get to know her? All that
Yeah.
stuff was going through my head, and
Yeah.
and so it was very scary, and then I just kind of focused on, okay, here's a treatment plan. We can so I went through a chemo course, and it was I think they gave me like 3 courses of one type of medication that was a little more aggressive, and then there was this a newer immuno version of chemo that actually worked remarkably well, and I had 3 more courses of that.
Yeah. Yeah.
And and so that for me, it was just trying to get well, and I tried to focus on sort of small things, winning small things like walk around the floor, a couple times, keep walking, get some more, moving, stuff like that.
It's amazing how it focuses your attention, and something like that. From what
I zoomed in. I was like, let's just go a step at a time, right? And let's get, and try to get well. And then I started, I was hoping that it would work, and I was going to do everything I can to try to make that happen. I started doing a lot more exercise after that, because I was, because of course the first course of medication, it's like, well, one of the risk factors is it can hurt, it can damage your heart. So I'm like, well, let's make sure that I do everything I can to preserve that.
Yeah.
So it kind of shifted my priorities a little bit. I was like prioritizing my health, prioritizing my kids. During there, I was doing some work in the hospital too, because I had my computer and there's that, not much else to do. And that helped me kind of focus a little bit, but I think that another big thing I was like, I'm a Christian, so I had sort of drew in my faith a lot during that time as well. But, I also realized that things that I'm scared about aren't really so scary anymore. And so in particular, I've always been a little bit shy. And I realized, I think after that life is too short. You just have to have those discussions or do these things, and it's not so scary. It's, resolve it, and that kind of stuff. That was, I think, a big shift for me to realize that. And maybe that's part of kind of how things changed, going forward. And I think it was a couple years out, things were looking, a lot better. I had a couple setbacks. Like I had, after the first few treatments, I got a pulmonary embolism because of all the cells kind of in my system. That was not fun. That was not fun. Yeah, it was very, very. I was the one in the ER screaming, because I was in extreme pain.
Oh my gosh!
So, I made it through that, and thankfully I'm back, and I'm playing tennis and all this stuff, regularly and stuff. And I also kind of changed, in terms of my kids. I was like, I prioritized my relationship with my kids and with my family. I started taking trips with my kids. Like, I would take them to a conference with me or something, and I'd take one-on-one, so I would be able to have some time with them.
Yes.
And, just cherish the time more than I had, right, in terms of the time that we have. And so that was kind of another thing that sort of came out of that.
Yeah.
For me. So, yeah.
Yeah, that's actually right. That's truly inspiring. I mean,
I know you do that as well with I've seen you with your kids and your trips and things like that. So
I do my best. I always feel like, it's funny with kids. I mean, as you probably know, it's like you always feel like you're doing what you can. It's never perfect, and it's always you got to like decide whether it's like enough or you can always feel like you can do more. It could be better, and so it's a tough balance. And it seems though that I do that as well.
You don't have teenage girls, right?
No, I have no girls, just all boys. And
that's what we're in that phase right now. And man, oh boy. So
and they're all different too. I mean, and they're all different.
Oh
in the
yeah.
sense that, my youngest, you try to connect with them, and now they're kind of in their own little world, and they're happy. They're very self-sustainable. And you have to first disrupt that, and then make a connection, and then eventually they appreciate it. And
yeah, absolutely.
But I really, and also that's what I noticed is that, and I didn't know beforehand, but I was inspired by, obviously, and these sort of things have a life of their own. I mean, it's like it was just wonderful that you got better, and it was probably a combination of many things. But But I think it does do something. Like you said, you reset your priorities.
Yeah.
You're not scared by anything. You try to focus on what's important and decide what's important. And
Yeah. For me, it was like, I was like this, I can either just be defeated, or I can see what is the opportunity here. What is this telling me? How can I, benefit from this going forward? What changes can I make? So, yeah.
Yeah, and I really have noticed that at least since then. I mean, you really are, one of the most on paper and in real life in terms of impact. Been so impactful on the field. I mean, your metrics of publication are through the roof, and it seems that you have this, what some people could say, oh, well, he has a big group, but I mean, it's one thing having a big group, but also being able to impact each person. And I think what you've done as well is with your family too is sort of, you've learned how to sort of like slow down and focus on the most important things. And in some way, indirectly, that sort of allows you to be more productive. I mean, it's sort of a paradox in some sense, but and I think it's wonderful, and it's too bad. It's not too bad, but I mean, it's unfortunate you had to go through this, and you probably already had this. Obviously, you already had this sort of perspective to some degree beforehand, and it just sort of helped focus it even more. I'm always trying to keep a balance as well, and
Right.
It's hard. It's hard in science and work and family and everything.
It really is. Yeah.
Yeah. And maybe I'll ask to continue on with that. I mean, I think, and also I think part of the reason why I wanted to bring this up, that there's so many people in the field that are suffering various illnesses, and that they're not to feel alone. I mean, it's a struggle, and
I mean, even in just my group, I've, and you probably as well, right? Everybody's got their own individual set of things, and sometimes they're very challenging. There's seasons that people go through, and you see this, I think, at the larger scale as well. But
yes,
I guess it has kind of helped me also to kind of appreciate that more.
Yeah.
Also, yeah.
Yeah, in others as well, right? And not
Right.
just
Right.
yeah, I totally agree with you. And so yeah, that's one thing actually. I mean, realistically, I've been visited your group, and it seems like you have so many different things going on, and it seems like you have an influence on so many different things. It's probably all consuming in some way to do it well, but it's fun. I'm sure you're having a lot of fun as well. So.
Yeah, it's a lot, but I think there's a lot of good people that I work with, I think, which has, been helpful. And I think for me also, I tend to try to be very collaborative, and I try to encourage others to do that as well. Cause that's going to bring lots of benefits, because you learn more things from other perspectives, and they may reach out to you and want to do some work that you can contribute to. And, I think the other thing too, that, and I know this for my own, because when I first started in research, I was really struggling a little bit with productivity. And my first paper is not published. It's sitting in my laptop here. And it's an incredibly detailed, long paper that never got published. And, it's, I sort of realized later how to navigate that space in a way that would, so you want to be, you don't want to just produce garbage. You want to, but there are choices you can make to navigate in interesting ways, questions, that you can answer certain questions, and you can decide at what level of depth you want to do that. And match your claims to that level of depth, so you're not overclaiming or underclaiming. And so that can be a rich space. And that's I think that's kind of one of the things that I sort of learned. And then the other is, when I'm working with other people in my group, everybody's a little bit different, as you mentioned, and they have different skills and they have different things they like. And you put people on a project and they'll sit there and they'll just struggle. You put them on a different project and they'll be like, boom, and they'll just like start launching. And so sometimes you just have to reevaluate, the fit of the person to the project. Some people are more, algorithm development people. Other people are a little bit more on, let's try to make these things work so that other people can use them, and so it's finding that sort of impedance match between the person and the project to optimize your, help everybody else be as productive as you can. And so I think that's an important, point as you I'm sure know. So.
Yeah, that's actually really well put. Impedance matching, right? What they do. Yeah, and even I really like your insight in terms of matching the claim to the data with the paper. So people don't usually have, the claim they always feel is somehow implicit, and then they try to make the data as profound as possible or as perfect as possible. And which really, no paper is like that. And you have to just sort of try to, match that. And if you feel that it gives enough of an insight or new information, then it's good to go.
It can be useful. Yeah. So it's, to me, because I can be like, see, we did that. Look at this. This tells us something, and there's the next thing we're going to do to build on that, right? So it's like a progress report.
Yeah,
right?
no, exactly. And keeping people as they're doing things to sort of, kind of start thinking of a paper in terms of, what do you want to say about this,
Mm-hmm.
instead of just endless exploring or endless testing and verifying. That's amazing advice as far as that's concerned. All right. Well, maybe one last question. So, we covered almost everything. I mean, you gave great advice. You sort of projected a little bit of the future. Any other parting thoughts in terms of advice for, people starting out as like an assistant professor or something like that,
Mm-hmm.
and are aspiring to build a big group or to find a good chunk of a field to delve into?
I guess, okay, so I've got a few points I can highlight. One would be just remember that people that you interact with will probably be your colleagues for many years to come. And so it's not worth having this little spat over little things, right? Because you will work with them many more times. You will meet them many more times. So think about the bigger perspective there.
Yeah.
I've been with my wife. We've been, we study, parenting and things like that. So there's this context of relational parenting that we've been doing recently, which is, don't focus only on the immediate, but focus on the long-term relationship. You want your kids to want to spend time with you when they don't have to, when they're older, right? So that kind of thing. To translating that to research, we should be doing relational research, right? So you should be thinking about relational, collaborative kinds of things, not selfishly about just your own stuff. I mean, you have to move your things forward, but that can become pathological.
Yes.
And if you do that, it'll help you build a larger team, because other folks that you work with will be like, later on, they'll be like, oh, I had a pleasant interaction with that person. They might have some insights. Let's reach out to them. And, that they're more likely to do that if they had a good interaction with you.
Yes.
I would say, based on all my interactions with my students, you don't have to bash existing approaches to highlight the advantages of your new approach.
Yeah.
I think that's the sort of beginner mistake. The rookie mistake is like, that's bad. Look at my great approach. It's so much better. That is transparent, and it's kind of, just boring, I guess, in some ways, right? So you discount it. Everybody discounts it when they see that.
Yep.
I think the more you don't have to make extreme statements about your work. Everybody wants to, but I think the more extreme statements you make, the less correct they are.
Yeah.
Typically you can draw attention to your work creatively. Be creative without being a jerk.
Yeah.
You don't have to, just say really rude things all the time and highlight your work. And, I think look for something that existing approaches might be overlapping. Where are the gaps?
Yeah.
The simple story that you hear is often not the complete story. It's loaded with choices that were made, or just to look at it from a particular perspective. But there are probably other things that were not looked at, and those might be right for future study. So those are my few points.
Those are great. Those are that is awesome. I'm going to have to re-listen to this and write these down. Put in the show notes. But
you can use a few of those if you want.
Yeah. Well, thank you. I think, and I think you're right. I think overall, I mean, just having it's not a zero-sum game, and it's all about relationships right. That's so important.
And I don't mean to convey that I've done this perfectly, because I haven't. I mean, I'm speaking from don't do what I did kind of perspective, right? In some ways. So, yeah.
Yeah. I mean, right. Even if we know that, you can sometimes make mistakes in different ways, but
Right.
And it's always somehow right. I mean, somehow it's always tricky, because when you see something as like incorrect, it's like you're so it's so satisfying on some level just to identify that.
Mm.
And but you have to do it in a way that's sort of right, like you do, like like you say, just in a more collegial way to realize, it's just something you're trying to help. It's not like you're trying to it's not a zero-sum game. It's sort of, everyone's trying to learn. And
Yeah.
great advice, and great. We barely scratched the surface of what you did, but I just wanted to try to get the highlights a little bit. And I look forward to seeing your next papers as they come out. And I can't quite read fast enough to keep up with them coming out, but I look forward to it. And you're having a huge impact. So thanks.
Well, thanks for the opportunity to this has been fun. I've enjoyed it. So.
Yeah, so have I. Thanks.